Imagine for a moment that you could track exactly 6,438 siblings for their entire lives.
Right, every single one of them.
Yeah, you monitor every single detail of their existence, you record exactly when they pass away, and you have like complete unrestricted access to their genetics. Your goal: to decode the hidden pacemakers of aging, to find out if there are biological timers ticking away inside of us dictating the length of our lives, and whether those timers change based on our age or, well, our sex.
It honestly sounds like a crazy thought experiment, but it is actually one of the most rigorous biological undertakings in recent history. We are looking at a stack of incredibly detailed research papers today. They're all centered around a 20-year study on a highly specific group called the UMHET3 mouse cohort.
20 years. That's massive.
It's unprecedented. And the mission of our deep dive today is profound. We want to understand the genetic, the molecular, and the evolutionary trade-off that shape lifespan. And ultimately, we want to look at the concrete evidence proving how these exact findings translate directly to human longevity.
Okay, let's unpack this, because the sheer scale of the research in these sources is just mind-boggling. Most of the aging studies we usually see take a pretty simplistic, almost lazy approach.
Yeah, the classic lifespan study.
Right. They essentially just look at a population, wait until everyone passes away, and see who lived the longest. It's like only watching the final mile of a marathon to understand how the entire race was run. You completely miss all the strategy. But the researchers in this study, they did something completely different. They used an algorithm called actuarial mapping, and that lets them look at the entire lifespan step by step.
Which completely flips our traditional understanding of longevity on its head. I mean, the traditional search has always been for one magical aging gene that guarantees a long life, like a switch you can just turn on.
Like the fountain of youth gene.
Exactly, but actuarial mapping shatters that idea entirely. Instead of just looking at the final age of death, the researchers studied 72 nested survivorships.
Nested survivorships. Let's break that down for the listener.
Sure. So to picture this, imagine taking the population's entire lifespan and slicing it into distinct 15-day intervals.
Right, and they start tracking this at day 42, which is basically puberty for a mouse. That is the baseline where the biological clock officially starts ticking for the study.
Yeah, from that 42-day mark, they move forward in those 15-day steps all the way to 1,100 days old.
Which is incredibly old for a mouse. Right?
Oh, it's extreme old age. To put that in perspective, only the oldest 8.7% of the mice even make it to that 1,100-day mark. By looking at the population in these truncated 15-day slices, the algorithm allows scientists to see exactly when a specific gene variant becomes an advantage and when it suddenly morphs into a deadly liability.
I really want to latch onto that concept of a gene becoming a liability, because it fundamentally changes how we think about our own DNA. Think of your genetic code like a reality TV elimination show.
Oh, I like that analogy.
Yeah, so in a reality show, different survival skills keep you safe at different stages of the game. Some genes might get you voted off the island in the very first round when you were in your youth. Other genes might do absolutely nothing for you the entire season. They just sit there dormant. But they are the exact unique traits that save you in the finale when you reach old age.
That is an incredibly accurate way to visualize the mechanism at play here. By running this actuarial algorithm, the researchers combed through the entire genome and discovered 29 distinct regions, which they named Vita loci.
Vita, as in the Latin word for life.
Right, Vita loci. These are specific genetic zones that act as the master controllers of lifespan, and the data shows their effect sizes swing wildly depending on the time slice we're looking at. We are talking about specific gene variants causing up to a 15-day difference in life expectancy over just a 45-day period in the mouse's life.
Wait, I have to stop you there. So a gene doesn't just make you live longer overall, it acts more like a...
Like a timed explosive.
Yeah, a timed explosive. That seems so counterintuitive to everything we are taught about having quote-unquote good genetics.
It really does challenge the conventional wisdom, but the biology relies heavily on your exact age. We can actually break these 29 Vita loci down into 4 distinct categories based on when their specific biological timer goes off.
Okay, let's go through them.
First, we have the durable loci. A prime example from the research is Vita 1a. This is the closest thing to what you just described as traditional good genetics.
So it's just universally helpful.
Exactly. If you inherit the beneficial version of Vita 1a, its effect is persistent. It continuously works to lower your mortality rate, whether you are a young adult mouse or a senior mouse.
Okay, so a durable locus is the steady workhorse. It's always helping. But what about the ones that shift?
That brings us to the second category, the diminishing loci. You can think of these as rate of aging modulators. Take the Vita 14b locus, for example. Early in life, the difference in survival between mice with the beneficial variant versus the harmful variant is massive. The good gene provides this huge shield.
But I'm guessing that shield doesn't last.
You're right. As the mice get older, those survival curves gradually converge. The biological protective effect literally wears off over time. It leaves the older mice equally vulnerable regardless of what variant they have.
So the shield just degrades. Wow. But the third category in the research is what really threw me, the transient loci.
Yeah, the transient loci are fascinating because they operate strictly within a specific window. Consider the Vita 1c locus. This gene only acts on survivorships that include adults younger than 500 days.
So it operates strictly in midlife.
Strictly midlife. If you manage to survive past that midlife point, Vita 1c essentially turns off. The cellular pathways it regulates just stop affecting your mortality altogether. It is a true midlife specialist.
And that perfectly sets up the final category, right? The late-acting loci, like Vita 9c.
Huh?
From what I read in the sources, these genes do virtually nothing for the first 2.5 years of the mouse's life. They are completely silent.
Not a single blip on the radar.
But once the mouse reaches extreme old age, suddenly Vita 9c activates. It becomes a massive determinant of whether that organism survives another month. It's that reality show finale gene I was talking about.
Exactly, which highlights a critical mechanism. Genetic influence is not static. But understanding when these genes act is only half the puzzle. We also have to look at who they affect, because all of these dynamics shift entirely when you separate the population by sex.
Okay, we're about 5 minutes into this deep dive, so let's do a quick mini recap so we don't lose the forest for the trees.
Yeah.
Because this is dense.
Good idea.
Bullet point 1. Actuarial mapping tracks survival in stages, not just at the end of life. Point 2. The researchers found 29 Vita loci that control lifespan. And point 3. Gene effects are essentially timed to go off at different ages, like that reality show elimination.
Perfect summary.
So let's pivot to the who. Let's dive deep into the sex differences, because the baseline gap in the data is staggering. At 42 days old, right at the start of the study, female mice have an 81-day lifespan advantage over the males.
And 81 days in mouse years is a massive biological head start.
Yeah, it's huge.
It is a profound advantage, but the actuarial data reveals a really fascinating convergence later in life. While females start with that massive lead, by the time the population reaches 740 days old, which is well into their senior years, that advantage completely vanishes.
Wait, it just disappears.
Completely. The life expectancies of surviving males and females become identical at that point. The obvious biological question is, why does that gap close? And the answer lies in a mechanism called genotype by sex interactions, or GXS.
GXS. Let's use a specific case study from the sources to explain how GXS actually works in reality. The researchers highlighted the Vita 2b locus. They tracked specific genetic variants here, and they labeled them with letters like CH, BH, CD, and BD.
Right. And looking closely at the CH and BH variants of that Vita 2b locus reveals the core of the sex difference. If you are a young female mouse, inheriting the CH or BH genotype is fantastic news. It grants a significant lifespan advantage, keeping your mortality incredibly low through the first 2 years of life.
But for the males.
However, if you are a young male mouse, inheriting those exact same CH or BH genotypes is an absolute death sentence. It drives a massive spike in early mortality.
Think of this like a shifting seesaw. On one end of the seesaw is a young male, on the other end is a young female. The exact same genetic weight that lifts the female up into a long, healthy life comes crashing down on the male, completely crushing his life expectancy.
That's a great visual.
I have to play devil's advocate here, though. How can a single physical gene be a blessing for one sex and an absolute curse for another? Or good for a youth, but bad for a senior? Evolutionarily speaking, shouldn't a bad gene just be weeded out over millions of years?
That is the exact question evolutionary biologists ask. We don't just want to know how something happens biologically, we demand to know why natural selection allowed it to persist. This seesaw effect is a flawless illustration of a theory called antagonistic pleiotropy.
Antagonistic pleiotropy. It sounds like heavy jargon, but what does it actually mean?
The mechanism is essentially a Faustian bargain.
A deal with the devil.
In a biological sense, yes. See, evolution only cares about one thing, and that's passing on your DNA to the next generation. If a specific gene variant makes a male mouse highly robust, aggressive, and fertile early in life, allowing him to outcompete other males and reproduce successfully, evolution will strongly select for that gene.
Right, because he's winning the mating game.
Exactly. But the Faustian bargain is that the exact same pathways driving that early aggression and growth, like hyperactive testosterone signaling and rapid cellular turnover, they actively become toxic later. They brain the body out and kill the organism in old age. The Vita to B flip perfectly mirrors this bargain.
So biology is essentially saying, look, I will give you incredible strengths and reproductive dominance in your 20s, but the payment comes due in your 60s, and the cellular bill will be fatal.
The payment always comes due. And there is a second evolutionary concept shown in the data, the mutation accumulation theory. Remember those late-acting genes we discussed, like Vita in males?
Yeah, the finale genes.
Right. These genes only cause damage or provide benefits long after the reproductive phase of life has ended. Because the organism has already passed on its DNA, natural selection is essentially blind to what happens next.
Because nature already got what it wanted out of you.
Evolution simply packs up and leaves. It ignores those late-acting genes entirely, allowing random, sometimes chaotic genetic mutations to accumulate without any evolutionary pressure to clean them up. This is exactly why extreme old age is so genetically volatile.
It really paints a picture of nature just like walking away from the control board once we hit a certain age.
Yeah.
But the genetics mapped in this study don't stop at just lifespan. There's a whole second set of genes the researchers mapped. Aside from the 29 Vita loci, they discovered 30 Soma loci.
Yes, the Soma loci are critical here.
And these don't just control how long you live. They specifically dictate the trade-off between your body mass and your life expectancy.
The mechanism here is vital to understand. The word soma refers to the body itself. These 30 loci are the genetic architects of what biologists call the disposable soma theory.
Disposable soma. That sounds intense.
It is. This theory is built entirely on the concept of resource allocation. A biological organism only has a finite amount of bioenergetic resources to spend. You can either spend those resources building a larger, more physically dominant body early in life, or you can spend those resources on cellular repair and maintenance to ensure a longer life. You cannot physically do both.
So building a bigger body early in life literally drains the biological bank account that pays for cellular repair later.
Exactly.
And the data in these sources shows this punishes males incredibly harshly. If you look at the numbers, if you are a big, young male mouse with a specific genotype at the Soma 3D locus, specifically the CD genotype, you lose up to 17.9 days of lifespan for every single extra gram of body weight you put on.
To put the severity of that penalty into perspective, a typical mouse might only weigh 30 or 40 grams total, so an extra gram is a massive percentage of their body mass. And the female mice with that exact same genotype, they only lose about 3.4 days of lifespan per extra gram. The lifespan penalty for early physical growth is exponentially higher for males.
But why? I mean, why does nature penalize the males so heavily for being large? Especially when being large is often the exact trait they need to secure a mate in the wild. It seems like a cruel biological joke.
It does seem cruel, but the underlying causality ties back to how male bodies process stress. In mammalian hierarchies, early body mass in males is driven by testosterone and highly aggressive growth signaling pathways. These pathways force the body into a state of rapid cellular proliferation.
Just growing as fast as possible.
Right. And growing fast generates massive amounts of oxidative stress and metabolic waste at the cellular level. The Soma loci dictate exactly how efficiently the body handles that toxic waste. In males, the genetic drive to grow large completely overpowers the biological pathways meant for cellular cleanup. It leaves behind damaged proteins and DNA that drastically shorten the lifespan.
Okay, let's pause for a second mini recap. 10 minutes in. Point 1. Gene effects drastically differ by sex, like we saw with the Vita 2b seesaw. Point 2. Evolution makes those Faustian bargains, which is antagonistic pleiotropy. And point 3. The Soma loci dictate the exact cost of early growth. Big young males pay a heavy lifespan penalty.
Spot on.
Which brings us to perhaps the most intricate and honestly the most intimidating layer of this entire 20-year study, epistasis. Up until now, we've been talking about these genes as if they operate in a vacuum. You have a gene, it does a thing. But biology is a highly tangled network. Epistasis is the phenomenon where genes interact with one another, and crucially, how one gene can completely mask or alter the effects of another.
The network effect really changes everything.
To visualize how epistasis works, think of a master volume knob on a complex stereo system. You could have the most beautiful, intricate genetic melody playing on one gene. Let's call it Vita 9A. It's doing its job perfectly. But if you happen to inherit a specific variant at another gene, completely across the genome like the BD genotype at the Vita 1b locus, it acts like someone walking over and turning the master volume knob all the way to 0.
Muting it completely.
Yeah, that BD genotype at Vita 1b completely mutes the effects of Vita 9a. It doesn't matter what your Vita 9a gene is trying to do. It is functionally silenced.
And mapping those volume knobs led the researchers to a truly shocking revelation. When they mapped out all the epistatic interactions determining which genes were muting or amplifying other genes, they found that these networks are strictly segregated by sex.
Oh wait, segregated entirely.
Entirely. The genetic networks that determine male lifespan have almost zero overlap with the genetic networks that determine female lifespan. The biological wiring is fundamentally, structurally different.
Okay, I have to stop and push back here, because this is an incredibly fascinating web of mouse genetics, volume knobs, and seesaws. But we are not mice. We're humans. How can scientists realistically take a massive, complex web of Vita loci and sex-segregated epistasis in rodents and claim this tells us anything meaningful about human aging?
That is the most important question in longevity science. You can't just jump from a mouse to a human and assume it all works. The researchers knew this, which is why they applied a rigorous ladder of evidence, grading the data step by step to prove translation.
So walk me through this ladder. How do we get from a mouse in a lab to a human being?
Level 1 of the evidence is the molecular mapping in the mice themselves. They didn't just point to a vague region of DNA. They narrowed these massive genetic zones down to highly probable specific candidate genes. For instance, within that durable Vita 1a locus we discussed earlier, they isolated a specific gene called ATP6V1H.
And what does that gene do?
This gene acts as a master regulator of autophagy. Autophagy is basically the cell's internal recycling plant, where it clears out its own damaged components. And within the late-acting Vita 9b locus, they isolated several other strong candidates.
Okay, so level 1 is finding the exact molecular suspects in the mice. But then they move to level 2, which is where I get really skeptical, because they use nematode worms, specifically C. elegans. Hold on, how can a microscopic worm that lives for a couple of weeks tell us anything meaningful about human biology? Are genetics really that similar?
I know it seems like a massive leap, but C. elegans is a foundational staple in longevity research for a very specific reason. Their lifespans are so short that you can test complex genetic effects in days rather than waiting decades. And the biological pathways for basic cellular survival are remarkably conserved across evolution. A cell clearing out waste in a worm works very similarly to a cell clearing out waste in a human.
So the core machinery is the same, even if the vehicle is completely different.
Exactly. The researchers took the specific candidate genes they found in the mice and looked for the exact evolutionary equivalent genes, which we call orthologs, in the worms. They then used a technique called RNA interference.
Which is what, exactly? Break that down.
Sure. RNA interference is basically a way of delivering a set of custom instructions to the worm's cells, telling them to completely ignore and silence a specific gene. They wanted to see what would happen to the worm's lifespan if they turned the mouse-equivalent genes off. Since you can't ask a worm how it feels, their proxy for lifespan is motility. If the worm is still wiggling and moving vigorously, it is in a state of high health span. If it stops moving, it is aging rapidly.
And the results from silencing these genes were pretty wild, right?
They were phenomenal. When they silenced a specific worm gene called ACOS10, which is the exact evolutionary equivalent of a gene found in the mouse Vita 9b locus, the worm's health and motility skyrocketed.
Skyrocketed just from silencing one gene.
Yeah. Silencing this one gene boosted their health span almost as effectively as altering the famous DAF2 gene, which is arguably the most heavily studied longevity gene in all of biology. It was a massive validation that the pathways discovered in the mice were universally relevant to aging.
Okay, so level 1, we map it in mice. Level 2, we prove the mechanism works in worms. That brings us to level 3, the apex of the evidence grading, human data. Because if this doesn't show up in humans, it's just a really interesting quirk of animal biology. How do they prove this affects us?
To bridge that final gap, they utilized Mendelian randomization on human genome-wide association studies, or GWS data. Mendelian randomization is a brilliant statistical method. Instead of just looking for loose correlations like, say, saying people who eat blueberries live longer, which could be influenced by a million other lifestyle factors, Mendelian randomization uses nature's own genetic lottery. It looks at the genes you are randomly assigned at birth to figure out if a specific gene biologically causes an outcome.
They're essentially using our own natural genetic variations as a massive, real-world clinical trial.
Exactly. And they took human blood samples and looked at the human equivalent of the genes they had just
All
validated
right.
in the mice and the worms.
And they hit absolute pay dirt with a gene called APEH, which sits right inside that Vita 9b locus we keep talking about.
The APEH gene is fascinating. In human biology, APEH produces an enzyme that is deeply involved in protein degradation. It helps the body chop up and clear out toxic, damaged proteins before they can build up and destroy the cell. The researchers found that natural variants in the human APEH gene are directly causally associated with human parents living into the extreme top 10% of age brackets.
Wow.
Yeah, they tracked this through massive databases of human lifespans. The genetic architecture they mapped in a cohort of mouse siblings in a laboratory directly predicted extreme longevity in real human populations.
So, bringing this all together, we've gone from tracking 6,000 mouse siblings to uncovering timed genetic explosives to proving the disposable Soma trade-off where early growth damages late life repair, to validating those exact cellular recycling genes in human centenarians. If we zoom all the way out, what is the ultimate synthesis of all these sources?
The ultimate takeaway is that the traditional medical search for a single universal anti-aging pill is fundamentally flawed. Because genetic effects flip their polarities based on your exact age and your sex, any intervention in the future must be highly personalized and timed perfectly.
Timing is everything.
Exactly. For example, based on the Soma loci data, blunting body weight and metabolic growth pathways is absolutely crucial early in life, especially for males, to preserve those cellular repair mechanisms. But later in life, treating frailty requires completely different biological targets. You cannot apply a 30-year-old's biological intervention to a 70-year-old body. The genetic pacemakers have shifted.
It requires a complete paradigm shift in how we view health. And that brings us directly to you, the listener. We've just unpacked how your biology is executing a genetically pre-programmed series of trade-offs at this very moment. Knowing that the rules of your own biology change as you age, that what benefits you in your 30s, like building massive physical mass, might be the exact mechanism that generates the metabolic waste that harms your longevity in your 70s, how might you change your own lifestyle habits? Are you optimizing for the round you're currently in, or are you optimizing for the finale?
It really is a question of understanding which biological timer is currently ticking inside you.
I want to leave you with one final lingering thought to explore on your own. We talked earlier about how evolution is essentially blind to us after we reproduce. It packs up, walks away, and allows genetic chaos to accumulate. If that is true, if evolutionary forces completely stop caring about us after our reproductive years are over, are the latent life longevity genes we see in supercentenarians, those people living past 100, actually just total evolutionary accidents? Think about it. We spend billions trying to decode the secret to extreme old age, but maybe the ultimate finale of our biological reality show is nothing more than a random roll of the genetic dice. Keep questioning the science of your own biology.